Welcome to the video on drug substance particle size importance for the pharmaceuticals. Simply, this video is regarding why particle size of the API or drug substance is critical and is very important whenever you are working onto the pharmaceuticals. So let's start with the video. See, there may be the different formulations like tablets, capsules, maybe different route of administration like oral, topical, injectable, or inhalation. The formulations may be powders, pulmonary administration, inhalation formulations, topicals, oral formulations like tablets, capsules, and some formulations which are required to be inserted into the body cavities. Some formulations may be like suspensions or powder for making the oral suspension or powder for making the reconstituted injectable suspension. So the formulations where the drug is present in the solid form, that means it is insoluble form. For those formulations, particle size is very important. And for those formulations, the PSD, that is particle size distribution specification, should be included and should be designed very scientifically. So whenever you are working onto the formulations where drug is present in the insoluble form or in solid form, that time you should always have a very critical understanding about the particle size distribution. See, there are four classes of the drug substances based on BCS. BCS class 1, 2, 3 and 4. BCS class 1 involves the drug substances which have high solubility and high permeability. BCS2 drug molecules are having low solubility but high permeability. BCS class 3 drug substances are highly soluble but low permeable, and BCS class 4 molecules or drug substances are low soluble and low permeable. So mainly BCS class 2 and 4 are the classes where particle size of the drug substance will have much more impact. But the PSD limits are required to be set for all the BCS class drugs. It is not like that this API is highly soluble, so we are not going to fix the PSD limits. It is not like that. The PSD impacts many of the aspects of the manufacturing, stability, storage, handling, manufacturing process, and other CQs. So we will see those things in the upcoming slides. The PSD criteria or PSD or particle size is a very important aspect, or you can say it as a critical material attribute, and it is not only for the APIs, it is also for the excipients. But in this video we are only talking about the API PSD. So it is a functionality, you can say functionality related characteristics, FRC of the drug substance, and it is a critical material attribute of the drug substance. So critical material attribute means it affects the safety and efficacy, and that's why PSD is the critical material attribute. In the QBD, you have to, or you should involve this CMA as a part of the formulation development, and in the pharmaceutical development report you have to write the effect or risk assessment about the drug substance particle size. See, the PSD of the drug substance impacts the flow. If material is very fine or micronized, it will have less flow or it will be very poor flow, and if it is granular then it will have good flow. Density: the higher particle size material will be dense, but the micronized material will be less dense. Then micronized material will be difficult to handle, it may have static nature and dust generation during handling will be more compared to the drug substance with larger particles or the higher pHD or granular APIs. Then mixing nature or mixing behavior. Generally, the micronized materials will get mixed with the other materials or excipient in a better way compared to the larger particle size, larger particles of the API. But it also depends on to the mixing equipments, mixing time and other factors. But generally, the micronized material will mix better. Then impact onto the compressibility and compaction. When you talk about the oral solid formulations like tablets and capsules, that time particle size of the API will have impact onto the compressibility and the compaction behavior. It also depends on to the dose of the API or API load of the formulation. Then hygroscopicity. Generally, the drug substance or material, if it is having more surface area, it will absorb more moisture. So generally, it can be said that the micronized material will be more hygroscopic compared to the coarser particle size material. Then solubility. Micronized material generally are having good solubility compared to the coarser material or granular particle size API. Then degradation nature. As a general science, micronized material will be having higher sensitivity towards degradation compared to the coarser particles, because the low particle size or micronized material will have higher surface area, so their reactivity can be more, but it is not a general rule. It depends on to the formulation and it depends on to the molecule on case by case basis. Now coming to the impact of pHD of API onto the drug product CQS. So pHD can impact many of the CQS, and you can say you should do risk assessment for all the CQS. First is appearance or description. Generally, the color varies depending on to the pHD. It is also not same for all the APIs. Then assay and content uniformity. Plane uniformity and content uniformity is affected by the particle size distribution. Dissolution and drug release. Because particle size impact the solubility, intrinsic solubility, equilibrium solubility, and that's why the dissolution rate and drug release is governed by the pHD. Then impurity and related substances. pHD also have impact onto the impurity generation, impurity formation, and related substances. Also, this pHD also plays critical role in nitrosamine impurities. Stability of the formulation including the in-use stability. So whenever the formulations are made, that time the pHD impacts the stability, whether it is formal stability or whether it is in-use stability. Ease of administration and uniformity of dosing, specifically in case of suspensions. So whether it is oral suspension or suspension for injection. Then the critical formulation is pulmonary type of formulations which are required to be inhaled. The particles get inhaled into the respiratory tract and their deposition into the respiratory tract will be dependent on to the particle size. So these are the CQAs. Many other CQAs you can write down and study. Then effect of pHD onto the safety, therapeutic effect, patient compliance and the efficacy. As pHD affects solubility and rate of resolution or drug release, it affects the safety, efficacy and therapeutic effect. Generally, larger particles dissolve slowly and their absorption rate and extent by oral route will be dependent on to the particle size. For pulmonary administration and inhalation, formulation pHD affect the rate and extent of API in the respiratory tract parts and absorption into the bloodstream. Thus, the pHD of the API have impact onto the plasma concentration onto the Cmax and Tmax, and in turn, this impacts the safety and efficacy of the pharmaceutical formulations. The larger particles generally have less surface area, and that's why these particles may produce less irritation. Uh, these may produce more irritation to the GI tract because of their the larger particles, and these larger particles dissolve slowly and may take more time to clear from the GI tract. Smaller particles opposite nature will have, and smaller particles will have more surface area, will produce less GI irritation, and may get clear easily from the GI tract within a shorter time compared to the particles with larger particle size. So this impacts the patient compliance. As the side effects impacts the patient compliance, the formulations are required to be designed and manufactured considering the patient compliance. The PSD control is very important as the PSD impacts the safety, efficacy, and all the other CQAs. The PSD becomes very important, and this PSD is required to be controlled, and this is having a prime importance. The PSD limits are required to be designed very scientifically. Mainly 3-tier PSD limits are given considering the D10, D50 and D90. That is the D10 means the 10% of the particles, then D50 that is median particle size, and D90 that is 90% of the particles size. So I have made different videos on designing the particle size specification, and also I have made the video on how particle size is given. That is what is the meaning of D10, D50, D90, D100. So you can go through those video from this channel and you will have good understanding. See, as the PSD impacts the safety, efficacy, patient compliance, the regulatory bodies always ask about the PSD, how the PSD is studied, how the PSD is finalized, and what are the PSD specifications. The regulatory agencies or regulatory bodies, whenever they are going to approve your formulation, that time they ask about the science behind the PSD specification. They also require the data, how PSD affects the dissolution, how PSD is selected, what is the science behind selecting this specific particle size. So, whenever you are making a pharmaceutical development report, in that always include this information. So, this is regarding the drug substance particle size importance for pharmaceuticals. I have made other videos and these are available onto the Pharma Learning in Depth channel. So, I request you to go through those videos to have good understanding about the topic. Thank you for watching the video.